there, cherubs, and welcome back to another biology lesson. So today we're going to address question 2. So far we've talked about meiosis and how alleles segregate randomly into separate gametes, and this makes 4 haploid cells, which can then be passed on or used for reproduction to create offspring. But how the heck did we come to the understanding of all this stuff? Well, first thing I want you to do is predict the offspring between a tall and a short plant. What I want you to do is write down your prediction if you cross a tall and short plant. So pause the video and write down your prediction. Welcome back. So why do you think, just ask yourself, that there are no medium or short-sized offspring here? So we had 2 parents that were both one was tall and one was short, and then their offspring both came out tall. Kind of strange, but pause the video and see if you can hypothesize or predict as to why, even though we had a tall and a short parent, why did all of their offspring come out tall? Okay, so now this gets extra weird. So we've got one generation here with a tall plant and a short plant. And when those 2 were crossed, they both came out tall. But then these 2 plants were crossed, and we got 3 tall plants, but then 1 short plant. How the heck did that happen? Well, these are the exact questions that Gregor Mendel asked himself when he was doing his experiments with pea plants. Gregor Mendel is known as the father of modern genetics. And so far, we're going to address just a little bit of vocab. So in Mendel's experiments, he crossed many different types of pea plants. Sometimes he looked at height, sometimes he looked at color, sometimes he looked at pod shape, all that other good stuff. So in his experiments, he always started with a pea generation. And that's this generation up here. This is your pea generation. They're the parents. They're the first generation. Okay? So here we got the pea generation, the parents. Their offspring, between the parents, their offspring is called the F1 generation. They're the first babies. Okay? Now, if we take the plants from the F1 generation and we cross them, they're going to produce offspring as well. And the F1 generation's offspring are called the F2 generation. They're the second babies, the second offspring. So we go from the P generation to the F1 generation to the F2 generation. But as you'll notice, these results are confusing because you've got a tall and a short plant in your P generation, and all the plants in your F1 generation come out tall. Then if you start crossing tall plants to have babies, 3 of them came out tall and 1 came out short. It was a 3-to-1 ratio. So these results were confusing to Mendel because all of the offspring were tall. There were no short or medium in that F1 generation. So there wasn't like a blending of traits. But then the short trait reappeared in the F2 generation. So this is what Mendel studied in his time. Okay? He looked at all of these different crosses, and he started piecing together some very important vocabulary that y'all might have heard before. So versions of certain genes or alleles can be dominant or recessive. A dominant trait will always mask or overpower a recessive trait if it is present. So in this case, tallness would be the dominant trait because you seem to have more tall plants than short ones. Anytime the tall trait was present, it always masked the short trait. And a recessive trait will only be expressed if dominant alleles or dominant genes are absent. So we normally represent dominant traits with an uppercase letters. That would be like capital R, capital A, capital T. I just did not mean to spell rat, but we'll go with it. And recessive traits are usually represented by lowercase letters. Lowercase r, lowercase a, lowercase t. They are usually represented by lowercase letters, while dominant has uppercase. Okay, so up here we have different traits that Mendel looked at. We have a dominant trait that was purple versus white. So flower color, purple would be the dominant trait and white would be the recessive trait. Plant height, as we just saw, tall is the dominant trait and short is the recessive trait. Seed color, yellow is dominant, green is recessive. Seed shape, round is dominant, wrinkled is recessive. Pod color, green would be dominant, yellow would be recessive. Pod shape, inflated or full, would be dominant, and then constricted or flat would be recessive. And then flower position, axial would be dominant, terminal would be recessive. But the main one we're going to focus on here is going to be like the colors, the tall versus short, yellow or green seeds, that sort of stuff. Each individual has 2 alleles for a trait. And we talked about this. You get one from mom, one from dad. And they give you your traits. Okay? And these alleles can have different combinations. You could, let's go back to the dimples example that's in your output page. Okay, let's say this is coding for dimples. Okay? So if you have a capital D, not only is this dominant, but we also refer to this as homozygous dominant. Okay, because there are 2 of the same capital letters here. Now I come here, and this still is going to be the dominant trait, so no dimples, but it's heterozygous. And what I want you to do is compare homozygous versus heterozygous. In heterozygous, you have a capital D and a lowercase d. In homozygous dominant, you have a capital D, capital D. Now, if we come over here, we have homozygous again, only this time, this person is homozygous recessive, so they would have dimples, if dimples is the recessive trait. So they have 2 of the same. They're just homozygous recessive because they are both lowercase letters. So in order to figure out these combinations, I'd have to be specific and say a homozygous dominant father produced offspring with a heterozygous mother. What would their gene combinations look like? I would have to be specific about that. Okay? This is called a genotype. A genotype is the allele combination. Big R, little t. And that's going to determine somebody's phenotype or their physical traits. Phenotype starts with PH and physical trait starts with PH. So your genotype, let's say we're looking at dimples, whether or not we have those. Capital D codes for not having dimples. So if I am capital D, that's my genotype. My phenotype would be having no dimples. If I'm heterozygous, remember that dominant trait is still present. So if I'm heterozygous dominant, my genotype would be capital D, lowercase d. And my phenotype would still be no dimples. If my genotype was homozygous recessive, then I would have lowercase d. And in this case, my phenotype would be having dimples. Now, you are going to get a lot of practice with this. This is a lot of vocab at once, but I just wanted to get you acquainted with it so that you know that it exists. So these are examples of phenotypes in regards to AAP plant. So the phenotypes are all listed here. So here are all the physical traits up here, and they all are going to have a specific genotype or letters that are going to help represent their traits. And these are all of the different phenotypes that Mendel looked at. Okay. So dominant is always going to have an uppercase letter. Recessive is always going to be represented by a lowercase letter. Dominant is always going to mask a recessive trait. So these would both show the dominant phenotype, the dominant physical trait. This one would show, this genotype would show the recessive phenotype. Genotype is the allele combination. Okay, so we will review all of these topics on Monday when I'm back, but thank you for watching and have a great rest of your day.